Good morning and welcome to the Sermonix clinical update on ELAINE phase 2 clinical programs. At this time, all attendees are in a listen-only mode. A live Q&A session will follow today's formal presentations. If you'd like to submit a question, you may do so at any time throughout the webinar by using the Q&A text box at the bottom of your webcast player. At this time, I'd like to turn the call over to your host, Dr. David Portman, Chief Executive Officer and Chief Medical Officer of Sermonix Pharmaceuticals. Please go ahead, Doctor.
Thank you, Sarah. Thank you all for joining us today on our first analyst investor call, largely because this for the first time was a chance for us to show some of our data at the recent ASCO annual meeting. And given that we now can share some of this data with you in the public domain, we thought this would be a great opportunity to have several of our investigators who've been working on our program for quite some time, as well as some of our management team, join you and learn more about our exciting program around lasofoxifene, a novel next generation oral selective estrogen receptor modulator, the only one that's currently in the space being developed for targeting ESR1 mutations. Sarah Sammons from Duke University is currently an assistant professor of medicine at Duke and the Duke Cancer Institute. Her clinical focus is in breast medical oncology, and she serves as assistant director of breast cancer clinical research at Duke Cancer Institute and as breast leader for the Centers of Brain Metastases and the Cancer Immunotherapy Research. Her research focuses on the development of novel therapies for the treatment of metastatic breast cancer, and she's designed and implemented several institutional, national, and international clinical trials in advanced breast cancer. Following Dr. Sammons is Senthil Damodaran, MD, PhD. Dr. Damodaran is a medical oncologist, physician scientist with expertise in breast cancer, cancer genomics, and the development of novel targeted therapies. He serves as an assistant professor in breast medical oncology with a joint appointment in investigational cancer therapeutics at MD Anderson Cancer Center, and his focus is on genomic-driven clinical trials, applications of genomics for targeted discovery in breast cancer, and characterization of secondary drug resistance mechanisms. At MD Anderson, he led their department's genomic-directed clinical translational effort and is actively involved in several NCI initiatives. We couldn't be better represented here by these two outstanding clinician scientists. Let me go ahead and turn it over to Dr. Sammons.
Hi, everyone. Thanks for having me today. I'm going to be talking about a novel SERM, lasofoxifene, for ESR1 mutant hormone receptor positive breast cancer, a novel therapeutic strategy that I feel pretty excited about. These are my disclosures. So just to identify the problem, hormone receptor positive breast cancer is the most common type of breast cancer, and 281,000 women will be diagnosed with invasive breast cancer on a yearly basis. And in terms of breast cancer subtyping and the way we treat them, almost 80% of all new breast cancers are estrogen or hormone receptor positive. So the vast majority are hormonally driven. And there's currently more than 150,000 women living with stage 4 metastatic breast cancer in the US. Endocrine therapy, so therapies that block estrogen or modulate the receptor alone or in combination with other targeted agents, are currently the mainstay for treatment for advanced breast cancer. These treatments, at least in the beginning, are successful, but resistance inevitably develops, and they have side effects, including loss of bone density, which leads to increased fracture risk and diminished quality of life in terms of vaginal, sexual health, vasomotor symptoms. So they're a little bit difficult to tolerate. This is currently what the average treatment course of a woman with advanced or metastatic hormone receptor positive breast cancer looks like. Patients are usually initially started on an aromatase inhibitor with a CDK4/6 inhibitor in the first-line setting. If they're what we consider endocrine sensitive, so meaning that they either have not had endocrine therapy before or they've relapsed within later than 12 months after their adjuvant therapy, they inevitably develop resistance on that after about 24 months. And then at that point, we don't get progression-free survival intervals nearly as long as that. And we usually try a second endocrine therapy with a targeted therapeutic. That usually lasts 5 to 8 months, and then resistance developed. And then we sequence chemotherapies and antibody drug conjugates after we've exhausted our endocrine and targeted therapy options. But resistance to all of these therapies inevitably develop. We don't have a cure, and new treatments are needed. One of the major resistance mechanisms that we see in hormone receptor positive breast cancer is an acquired mutation in the estrogen receptor. And these are called ESR1 mutations, and they generally form in the ligand binding domain. And we know from many clinical trials and translational studies that ESR1 mutations lead to endocrine resistance. So the first two figures on the top, these are the most common types of ESR1 mutations we see. We often see Y537S, Y537N, and C. And then the most common mutation that we see is D538G. These are most often monoclonal, but about 25% of the time they're polyclonal. And we know from many series that ESR1 mutations breed resistance to aromatase inhibitors, and some of them even breed resistance to fulvestrant, which is the current only approved selective estrogen downregulator. ESR1 mutations are not common. You can see on the bottom left, the bar graph, they're not common in patients that get aromatase inhibitors only in the adjuvant setting. We only see them happen at this point about 4% of the time. When a patient originally relapses with breast cancer, even if they've metastatic, even if they've had an adjuvant AI, only about 8% of them in the first line setting have ESR1 mutations. But where we really see ESR1 mutations develop is after first line aromatase inhibitor therapy with a CDK4/6 inhibitor. And about 40% of patients with advanced hormone receptor positive breast cancer will develop an ESR1 mutation after first-line therapy with an aromatase inhibitor and a CDK4/6 inhibitor. There are currently no approved treatment options for patients with ESR1 mutations. So why do we care about these mutations? What they essentially do is they cause constitutive activation of the receptor, which what that means is the receptor is no longer dependent on the ligand estrogen. So we can't just block estrogen and prevent the cancer from growing. The receptor is constitutively activated regardless of blocking estrogen. So that's what the mutations do. And in doing that, they're resistant to aromatase inhibitors, and most are also resistant to fulvestrant. We know that patients with ESR1 mutations have shorter progression-free survival and overall survival on standard therapies. Currently, fulvestrant is the only gold standard therapy that we have for patients with ESR1 mutations because it works better than aromatase inhibitors, but fulvestrant still does not work very well at all in ESR1 mutant breast cancer. The progression-free survival of fulvestrant in very recent trials in the last year or two shows a progression-free survival of about 2 months with fulvestrant in ESR1 mutants, which is pretty lousy. So there are other oral SERDs out there, oral selective estrogen downregulators that are out there looking to optimize this progression-free survival. Most of them to date, which I'll show you, have been a bit underwhelming. So there is a high need for new therapies for ESR1 mutant breast cancer, and this is why we're excited about lasofoxifene. The anti-estrogen new generation space is very crowded. But it's becoming less and less crowded by the month. These are all the anti-estrogens that are out there in development. You can see that most of them are oral SERDs. Lasofoxifene is the only SERM that's currently in development for ESR1 mutant breast cancer. And then there's a variety of sort of new generation platforms, PROTAC, CIRCAs, serins. The only oral SERD so far to meet their primary endpoint in a clinical trial was elastrozant. And you can see in the intention to treat population with elastrozant after CDK4/6 inhibitor in the second line setting, the median PFS is still 2.7 months versus standard of care, which most of them got fulvestrant, was 1.9 months. So this is a high need population. This is the only positive trial, and it's pretty underwhelming. We have seen a couple other oral SERDs fail in this space already. We've seen giredestrant not meet its primary endpoint. We've seen amcinestrant not meet its primary endpoint. And there are some other trials underway. One of the main criticisms of these trials is that they did not select for ESR1 mutants, which we have done with lasofoxifene in its development. So what is lasofoxifene? We think it has more favorable profile in terms of pharmacokinetics and pharmacodynamics than other SERMs, and it's certainly differentiated from the investigational oral SERDs that are out there. It has excellent bioavailability. It has excellent volume of distribution, meaning it gets into the tumor. It has a long half-life of over 6 days. In terms of pharmacodynamics, another important thing about lasofoxifene is it's not only it doesn't only engage an ESR1 mutant receptor, it also engages and treats a wild type receptor as well with high potency and affinity. We also know that the 5 milligram dose that's been selected occupies and inactivates both wild type and mutant ESR1 receptors. Another thing that I'm excited about the drug is that it has extremely well characterized safety and tolerability. This drug has already been used in tens of thousands of women because it was developed previously as an osteoporotic drug. So we already have experience with it. And it's very rare that I can say a drug actually helps a woman do anything, but this drug actually improves bone density and treats urogenital atrophy. So it actually might improve, it does improve bone health, and it most likely improves women's vaginal symptoms. And we know that it's safe and it's well tolerated. So lasofoxifene preclinical and clinical data has supported its phase 2 fast-tracked program in ESR1 mutant advanced hormone receptor positive breast cancer. The reason for this is that there's a large amount of safety and efficacy data already from a non-oncology program. And it was fast-tracked in 2019 based on robust preclinical data that I'll show you in a minute. The phase 2 ELAINE-1 trial studied lasofoxifene in ESR1 mutants and enrolled its first patient in September 2019. It has completed enrollment and is submitted as a late-breaking abstract to ESMO 2022. And ELAINE-2, which my colleague will talk to you about, was presented at ASCO 2022, just a couple weeks ago. So this drug is being repurposed for ESR1 mutant breast cancer, but it's previously been studied looking at osteoporosis treatment. So one of the largest studies that it was looked at was an 8,500 patient PEARL study to look at its efficacy in treating bone loss. And what this study also showed is they also looked at the breast cancer incidence, the incidence of women developing breast cancer who got lasofoxifene. And lasofoxifene clearly reduced the risk of patients developing breast cancer. It also treats osteoporosis. So this, in a sense, makes me excited to put this drug in the adjuvant setting eventually. But there's clearly some activity there in terms of treating breast cancer, preventing breast cancer. The way that this drug was really discovered to have efficacy in ESR1 mutant breast cancer was in a lab at Duke University, actually. A postdoc in Donald McDonald's lab basically put these ESR1 mutations in vitro breast cancer cell lines and she used pretty much every endocrine therapy known to man to see which drug was going to have the most efficacy in ESR1 mutants. So she transfected these ER-positive cell lines with the most common mutants, Y537S and D538G. And she looked at the efficacy, the IC50s of different SERMs and SERDs. So raloxifene, fulvestrant, tamoxifen, elastrozent, which is RAD1901. And what you can see on the left is that all of the other SERDs and SERMs out there have reduced efficacy in the ESR1 mutants compared to wild type, whereas lasofoxifene does not. It has the same efficacy in vitro in the wild type and the two most common mutants. They also did a really interesting experiment where they looked at the allelic fraction, so the amount of receptors that were positive for wild type versus mutants, and they drove it up. So in the middle, basically all the way to the left is 100% wild type allelic expression. On the left is 100% ESR1 mutant expression, and in the middle is 50-50 mix of wild type and mutant. And it didn't matter what the allelic fraction of wild type versus mutant was, lasofoxifene had activity throughout, which was not the same once you drove up the mutant expression for fulvestrant. Lasofoxifene has also been studied in preclinical in vivo models. So this is a group that looked at lasofoxifene in a Y537S, which is supposed to be the most endocrine resistant mutation in MCF7 xenografts. And essentially what you can see is that lasofoxifene worked well in terms of tumor suppression in these models. And it also worked differently than palbociclib. And activity was seen with fulvestrant and palbociclib, but was essentially driven by the palbociclib and was not as effective as lasofoxifene alone. Efficacy was also observed with lasofoxifene and palbociclib, and probably was predominantly driven by the lasofoxifene. Dr. Demarandan is going to talk to you about ELAINE-2 and the significant activity of lasofoxifene with CDK4/6 inhibitors, and also in the post-CDK4/6 inhibitor space, which is very important. This is the same group. They just used these xenograph models to show that lasofoxifene inhibits in vivo liver, lung, bone, and brain metastasis and was superior to fulvestrant. So ELAINE-1, which is a study that I was involved in, was an open label, randomized, multi-center, two-arm signal finding study that compared lasofoxifene to fulvestrant. Patients had to be postmenopausal or they could be premenopausal, but they had to have their ovarian suppression with a GnRH analog. They had to have an ESR1 mutation present on circulating tumor DNA liquid biopsy that was confirmed centrally, and they had to have had progression on first or second line hormonal treatment, including an aromatase inhibitor and a CDK4/6 inhibitor. This was largely, this trial largely enrolled a second line population after aromatase inhibitor and CDK4/6 inhibitor. The primary endpoint was progression-free survival. Patients were randomized 1-to-1, 50 patients received 5 milligrams of oral lasofoxifene daily, and 50 patients received 500 milligrams of fulvestrant at its standard dose. We can't, I can tell you the baseline characteristics of the patients in ELAINE-1. It's fully accrued. It's submitted to ESMO 2020, but the efficacy results are embargoed. So I can't tell you much about efficacy today, but essentially the patient characteristics are matched throughout in terms of age. A large proportion, 2/3 of patients had visceral disease. 100% of patients in this trial had an AI and a CDK4/6 inhibitor. Something unique about this trial is they did stratify for Y537S mutation So we have a very large portion, 40% and 47% of patients that have this mutation, and 100% of the patients have an ESR1 mutation. The only results that I can share with you today is that for the first time with an endocrine monotherapy in an ESR1 mutant patient was a complete response noted. So one patient in ELAINE-1 had a complete response to lasofoxifene. She's a 63-year-old female with metastatic breast cancer to the pleura, the lymph nodes. She had had almost 5 years on palbociclib and letrozole with a good response, and then had progression in her pleural area. She was enrolled in ELAINE-1 and randomized to lasofoxifene. And currently at 72 weeks, she has a confirmed complete response by blinded independent review with 60-week duration of ongoing response. Her target pleural lesions had a sum diameter of 3 centimeters, and she had multiple non-target lesions that decreased as well. And they're essentially, her target and non-target lesions are essentially absent at this point. Her circulating tumor DNA at baseline, she had a D538G mutation and a Y537C mutation, so she was polyclonal. And at 72 weeks, her circulating tumor DNA is no longer expressing these ESR1 mutants. So we are seeing circulating tumor DNA decline with lasofoxifene. So I think that, first of all, ESR1 mutant hormone receptor positive advanced breast cancer is a strong area of unmet need. It's 40% of all of these patients. We do not have an FDA approved drug in this indication. And what we have works for 2 to 3 months, which is not great. In ELAINE-1, from what I can share with you so far, we have 1 reported CR in a hormone receptor positive patient with polyclonal ESR1 mutations to endocrine monotherapy, which has not been described. And the data will be presented at ESMO 2022. ELAINE-2, which my colleague will present, has very encouraging combination data with abemaciclib. The drug has a very favorable toxicity profile. And there's really just not much going on in this space. So we're really excited about the drug moving forward and the plan. And I'm gonna hand it over to Dr. Damodaran.
Thanks, Sarah. So I think Sarah did an excellent job in terms of laying the foundation for where we are in terms of ESR1 and the space being an unmet need, and also the properties of lasofoxifene in terms of targeting ESR1. So I'm going to be discussing the results from ELAINE-2, which is an open label phase 2 study of lasofoxifene in combination with abemaciclib for metastatic breast cancer patients who were treated with prior therapies and harboring an ESR1 mutation. So the results of this study was actually presented at ASCO a couple of weeks ago. And our study was also awarded the GRASP Advocate Choice Award, considering that to be one of the most upcoming promising studies. So these are my disclosures. And just to summarize in terms of how lasofoxifene works, as Sarah had mentioned, so lasofoxifene is a 3rd generation selective estrogen receptor modulator, or SERM. And studies have shown that it has excellent activity against ESR mutants, either as a single agent or in combination with CDK4/6 inhibitors such as palbociclib in comparison with fulvestrant. It works a little bit differently compared to the SERMs because it kind of modifies the constitutive conformation of the mutated estrogen receptor to an antagonistic conformation and thus inactivates the receptor. And compared to other SERMs like tamoxifen, it has excellent bioavailability as well as high affinity to mutated ESR1. So when we were looking at combinations, we realized that single agent endocrine therapies are unlikely to have a deeper clinical response. So we wanted a rational combination partner. So we picked abemaciclib along with lasofoxifene for this study for a variety of reasons. One is, abemaciclib is a well-known drug. It is approved for use in metastatic breast cancers, either as a single agent or in combination with endocrine therapy, and was recently approved for high-risk early breast cancers. And as you all know, like there are 3 CDK4/6 approved in the metastatic setting. So there's ribociclib, palbociclib, and abemaciclib. And even though they have similar progression-free survival, preclinical and clinical studies have shown that they are rather different in terms of their activity. And that is shown by the kinome profile here. So when you look at ribociclib and palbociclib, compared to abemaciclib, abemaciclib actually has a more wider activity in terms of targeting other CDK, like CDK7, CDK9, and it also has activity against other kinases, which we don't necessarily see with palbociclib or ribociclib. And preclinical studies from MDA and other institutions have actually shown that you still retain activity to abemaciclib when somebody has progressed on palbociclib or ribociclib. And Aditya's group at MGH had actually looked at abemaciclib or in combination with endocrine therapy or single agent, and they still observed a meaningful improvement in progression-free survival about 5.3 months in patients who received prior CDK4/6 inhibitor. So that brings us to the ELAINE-2 study. So basically this was intended as a primary safety study, but also look at efficacy of lasofoxifene in combination with abemaciclib. This was a multi-center study to include patients who had progressed on prior CDK4/6 inhibitor. We treated patients with lasofoxifene 5 milligrams daily and abemaciclib 150 milligrams twice daily, which is the approved FDA dose for combination therapy. And we treated patients until progression or withdrawal for any reason. As a safety study, the primary endpoint was looking at safety and tolerability. And we also looked at secondary endpoints, looking at clinical activities such as progression-free survival, clinical benefit rate, objective response, as well as duration of response. So these are our inclusion and exclusion criteria. We included all women who are 18 or older with metastatic estrogen-positive breast cancers. We allowed patients to have received at least 1 or 2 lines of endocrine therapy. We also allowed 1 line of chemotherapy in the metastatic setting. And more importantly, we enrolled patients with ESR1 mutation assessed by circulating tumor DNA. And for our study, we used Sysmex SafeSeq assay to enrich patients with ESR1. And we allowed patients with good performance status as well as adequate organ function. In terms of exclusion, we excluded patients who had extensive disease and concern for visceral crisis as they would require cytotoxic chemotherapy. Importantly, we also excluded patients with known inactivating RB1 mutations or loss, largely because for CDK4/6 inhibitors to work, you need an intact RB pathway. So that's one of the reasons we pre-specified this criteria. We also excluded patients with any clots like DVT or pulmonary embolism with the last 6 months due to concerns with potential risk of thrombophilia with the lasofoxifene, as well as abemaciclib. But we did allow patients who have been on anticoagulants for 6 months or more with stable clot risk. We also excluded patients who were on concomitant strong CYP3A4 inhibitors or inducers, largely because of interaction with abemaciclib. So this summarizes our patient disposition and screening. So we screened about 39 patients on the study, and out of that, 29 qualified. The first patient entered on the study into the study in October of last year, and we accrued quite quickly, and we completed all of our enrollment by June of 2021. Out of the 29 patients, there are 12 patients that are still on study and 17 discontinued either for progressive disease or because of physicians wanting the patients to come off the study or adverse events. This summarizes the baseline demographics and disease characteristics of patients in our study. As I mentioned before, we had 29 patients enrolled into this study. Majority of the patients were postmenopausal, about more than 80% were postmenopausal, and the median age of these patients, understandably, was about 60. And majority of our patients were white as well. And out of the 29 patients, more than 60% of our patients actually had measurable disease, and about 30% had bone-only disease. And importantly, more than 50% of our patients actually had visible disease, indicating disease in lung or liver and so forth. And about 50% of these patients also had polyclonal ESR1 mutations. And as Sarah mentioned, nearly 60% of our patients actually had the more aggressive variant of ESR1 called Y537S, were also enrolled in our population. This actually summarizes the prior therapies our patients had received on this study. About 50% of our patients actually received chemotherapy in the metastatic setting. And as you would expect, nearly 100% of our patients had received CDK4/6 inhibitor. And majority actually received palbociclib, which would reflect the practice patterns that we see in the United States. Interestingly, 4 patients also had received abemaciclib before. We'll discuss these patients a little bit more in detail later. And about 80% of our patients actually received prior fulvestrant. And 7 of our patients actually received either a PI3 kinase inhibitor or an mTOR inhibitor targeting the PI3 kinase axis. And 3 patients specifically had progressed on prior alpelisib. In all, our patients had received about 2 lines of therapy in the metastatic setting as a median. And the median duration of prior CDK4/6 for majority of our patients were about 2 years. And importantly, 80% of our patients were non-sequentially treated. So meaning that these patients had progressed on CDK4/6 inhibitors in the past, and they received some other treatment before they actually went on our study. This summarizes the common adverse events that were seen in more than 15% of our patients. As you can see, majority of our toxicities were more grade 1 or grade 2. And in fact, most of these toxicities could be attributed more to abemaciclib. Things like diarrhea, nausea, fatigue were the more common side effects that we saw. Effects on bone marrow, such as anemia and lymphocyte, would largely be more associated with abemaciclib, as is creatinine increase that is seen with abemaciclib. This actually summarizes the higher grade toxicities that we noticed on our combination study. So we've summarized the grade 3 and grade 4 toxicities. And as you can tell, we observed very little grade 3 or 4 toxicities with our combination. There's only 1 patient with grade 4 toxicity, and that was neutropenia, largely attributed to abemaciclib. But most of the other grade 3 toxicities were self-limited. We did observe 2 patients with thromboembolism on the study. One had developed DVT, and that was largely provoked because this patient had underwent knee replacement surgery before. And 2 of the patients also had pulmonary embolism, but these were actually incidentally noticed on surveillance CT scans, and they were not clinically symptomatic at all. And both of these patients were successfully treated with anticoagulants. They actually continued on our study with clinical benefit. As per protocol, we did not dose reduce lasofoxifene, but we did allow dose reduction for abemaciclib for toxicities. Out of the 29, 5 had dose reduction in abemaciclib, and they were dose reduced from 150 to 100 milligrams. No patient dose reduced to 50 milligrams twice a day, which is the lowest approved dose. And 4 of these patients actually had dose reduction because of adverse events such as decreased sodium, fatigue, nausea, or increase in creatinine. And 1 was dose reduced largely because of physician's discretion. So this actually summarizes the clinical benefit that we saw in our patients so far. The y-axis, the legend shows the biology as well as the treatments these patients have received in the past. And then x-axis shows the number of weeks that these patients are on trial. As you can see, like out of 29 patients, 12 patients are still on study, shown by these continued arrows pointing towards right. And on the y-axis, it tells you that we saw clinical benefit across different spectrum of patients. For example, we had 4 patients who were treated with abemaciclib, shown by the purple pentagon, showing significant clinical benefit, as is patients with residual disease, shown by amber circles, and patients with prior chemotherapy, shown by these dark triangles. And the dotted line shows patients who had stable disease at 24-week time point. And you can see like majority of our patients actually had clinically significant response with a clinical benefit rate of about 70% at 24-week time point. This actually summarizes the progression-free survival that we observed so far. Our progression-free survival was about 13 months, and out of which, out of the 29, 11 patients have progressed, and as I mentioned before, 12 patients are still on study. This actually summarizes the overall clinical response we saw in our study. Out of the 29, 18 had measurable disease, and out of these 18 patients with measurable disease, 9 had partial response, shown by the teal graphs pointing downwards. And additionally, 11 patients also had stable disease beyond 24 weeks. So in all, we had 20 patients who had clinical benefit rates that exceeded 6 months, so resulting in overall clinical benefit rate of about 70%. And overall response rate is about 50%. We seldom see such impressive response rates or clinical benefit rates in patients who had progressed on prior CDK4/6 inhibitor therapy, particularly with endocrine-based combination therapies. And the duration of response on a median was about 164 days, and the time to response was also largely similar. That was about 169 days as well in our study. This actually summarizes the response that we saw in patients who had progressed on previous abemaciclib. As I mentioned, there were 4 patients enrolled in our study who had progressed in abemaciclib before. And interestingly, 3 of these patients actually had clinically meaningful response. And this kind of summarizes the patient characteristics and the type of ESR mutation that they had at baseline and what happened to their allele fraction at 4-week time point. And you can see that in the 3 patients that had clinically significant response, we actually didn't detect any allele fraction at 4-week time point, which kind of indicates the response to ESR targeting agent with lasofoxifene. And one of these patients actually had partial response, and 2 of these patients actually had prolonged stable disease. 1 patient actually had progressive disease. And important to note that 3 of these patients actually received chemotherapy. So suggesting that the response that we see in this combination is largely attributable to lasofoxifene and not necessarily because of abemaciclib. And we looked at ESR1 circulating tumor DNA. In a majority of patients, about 90% of these patients actually had reduced or undetectable ESR1 allele fraction at 4-week time point, which kind of acts as a surrogate for clinical response later on. To kind of give you an idea of the response that we've seen in our study, I've summarized the clinical data that we have seen so far in the post-CDK4/6 inhibitor space. You know, Aditya and Seth Wander from Mass General had looked at abemaciclib either as a single agent or in combination with endocrine therapy for patients who had progressed on prior CDK4/6. And they had looked at PFS. They showed that the progression-free survival was about 5 months. And when you compare that with our study, you see an PFS of about 13 months, which is about 3 times more than what was observed in previous studies. The MAINTAIN study was actually recently presented at ASCO that looked at maintaining patients on CDK4/6. So they basically switched patients from palbociclib to ribociclib and see if that would actually improve their progression-free survival. While that improved PFS with endocrine therapy combination to about 5.3 months, and you'll see that in patients with ESR1 mutation, when ribociclib was combined with fulvestrant, the actual PFS was abysmal at 3 months, and which is, you know, and when you compare that with lasofoxifene, we've observed like more than 4 times that response. In the Emerald study, Sarah had mentioned before, looked at elacestrant as a single agent in ESR selected population. The overall PFS was about 3.8 months. And to kind of give you a flavor of about other targeting agents in this space, the BELIEVE study looked at fulvestrant in combination with alpelisib, but they selectively enriched for patients with PIK3CA mutation. And the absolute PFS that was observed in the study was 7.3 months. And not to mention alpelisib as a drug is very hard to tolerate and is associated with significant benefits. So this kind of summarizes the impressive progression-free survival that we saw with this combination. So in conclusion, ELAINE-2 showed really good tolerability and excellent and meaningful clinical efficacy with lasofoxifene in combination with abemaciclib in metastatic breast cancer patients with the ESR1 mutations who had progressed on prior CDK4/6 inhibitor study. And to my knowledge, this is the first study that showed a significant improvement in progression-free survival at about 13 months. Particularly putting in context of patients who had seen prior CDK4/6 inhibitors, and half of the patient received chemotherapy, and with a response rate of 50% and clinical benefit rate of about 70%. And what is also notable is the fact that this combination is very well tolerated, right? So when you look at drugs or drug combination, you also not only look at the efficacy of the drug, you also want to see how tolerable it is. If the drug or drug combination could be very effective, but if it's not tolerable, patients are not going to take it. I think alpelisib or Piqray is a great example. The drug has excellent activity in patients with PI3 kinase mutant population, but as an agent is very hard to tolerate, and majority of the patients have to dose reduce or discontinue therapy. And as impressive as we have seen the response in this study, and of course we need larger studies to support this combination as an effective and potential therapy in this largely unmet population. Thank you. And I'll turn it over to David.
Thank you very much, Senthil and Sarah, for what I think is a masterclass in both acquired endocrine resistance driven by ESR1 mutations and the post-CDK4/6 landscape. What I wanted to do in just a brief company overview is introduce our stellar team and board, as well as our future very near-term milestones. I founded Sermonix along with my co-founder, Dr. Miriam Davidson Portman. We both have been involved in clinical trials for the last 20 years, largely in women's health. We acquired lasofoxifene, a drug that we studied in phase 2 and 3 trials as investigators, as well as advisors to Pfizer on endometrial safety. They shelved the drug once they acquired Wyeth, and at that time we acquired several Wyeth Pfizer experts in SERM clinical development, Dr. Barry Kalm, our Chief Scientific Officer, and Simon Jenkins, our VP of Clinical Operations. Additionally, knowing that we were going to be working in the endocrine resistant space, we were joined by Dr. Paul Plourde, who's here with us today to answer questions regarding our program. He has over 25 years of experience, largely developing the endocrine portfolio at AstraZeneca, which included our active comparator in the ELAINE-1 study, fulvestrant, as well as instrumental in their tamoxifen and anastrozole programs. We're also joined by Elizabeth Attias, who's been with Sermonix since the inception. Beth has significant experience in all aspects of biopharmaceutical portfolio planning and market assessment and commercial launches, and deep experience also in the competitive landscape around SERMs and SERDs from her work in that space. Our non-executive chairman is Dr. Anthony Wild, also with significant global pharma experience, helping us to guide our team as well as lead our stellar board, which some of them may be joining us on the call, but just by way of introduction, deep, strong experience in drug development, including Dr. Michael Friedman, former Deputy Commissioner of the FDA and head of City of Hope Cancer Center, medical oncologist, and Beth Garner, a very well-respected Chief Scientific and Chief Clinical Officer at multiple pharma companies. And then we have executive experience from Tony and Dick DeShutter, former CEO at Searle and Pharmacia. And then joining our board from a very supportive investor standpoint, Thomas Pfister, representing the Hans-Peter Wilde family office, our first major investor after several earlier rounds. And then our first institutional round led by Perceptive Advisors Ontogeny Fund. And with that, we were joined by Fred Calori and Ben Askew, both with significant experience and excellent advisors. Regarding our near-term milestones, which we feel our team is fully capable of executing on, as we have in the 2 studies that are reporting out, Elaine 2 already at ASCO and Elaine 1 at ESMO. We're well capitalized through early next year, but we are starting a process to fund our registration program through approval. And as you can see here, with our top line data, we're ready to engage with the agency with proposed plans to move the drug into the registrational approval process. We also have several compelling abstracts that are being put together, as was alluded to, the impact of lasofoxifene alone and with abemaciclib on circulating tumor DNA, which will be submitted to this year's San Antonio Breast Cancer Symposium. Because our future program will expand beyond the US, we'll be engaging with regulators outside of the country and prepare for our phase 3 program, which we hope to enroll our first patient by late this year, if not the latest early next year. So with that, I think we'll go ahead and we have plenty of time for Q&A where you can utilize the expertise of our 2 advisors here and investigators, as well as our team. So let me go ahead and see if open it up to questions. Let me pull up the Q&A. While I'm doing that, Sarah, if you or Glenn have the first question while I'm looking for that, go ahead and please ask that question.
Glenn, you're on mute.
Apologies. Certainly, David. So the first question that just came in is, what is the advantage and rationale for focusing only on an ESR1 population?
So let me, I think that Sarah alluded to the pitfalls of not looking at an enriched population. I think that was one of our key advantages, is that we know that endocrine therapies can work well in a general setting. We've seen that in Parsifal as well as other non-enriched populations. And given the activity of lasofoxifene, which we think could be best in class, focusing on ESR1 mutations made a lot of sense to us. Let me have Sarah perhaps comment a little further, and then Senthil as well.
Yeah, I would agree. You know, we know from several other studies that have compared SERDs to aromatase inhibitors and that have looked at the circulating tumor DNA, that aromatase inhibitors actually perform as well as SERDs and SERMs in the non-ESR1 mutant population. So it's really the ESR1 mutant population that has a high need of these novel strategies because they have constitutive activation of the receptor. So they really need the receptor to be modulated or downregulated, whereas non-ESR1 mutants, they still, ESR1 wild type patients still respond to standard of care endocrine therapies.
Yeah, I completely agree. I think David and Sarah pretty much summarized. I don't think we've, studies that we've had so far suggested that fulvestrant or even exemestane probably has comparable activity in patients with wild type ESR1, but we don't have any dedicated agents that have shown, meaningful clinical response in ESR1 mutations, which can be seen as high as 40% in patients who had, received prior aromatase inhibitor therapy. So this is a largely unmet population. So it makes sense to actually go after this.
And maybe I can ask Beth, she's done a lot of our market research and talked to clinicians around how they would feel about having a targeted therapy specifically for the ESR1 population. We certainly know that the current treatment algorithm, when you identify a PIK3CA mutation, directs clinicians to a a targeted therapy, even a very poorly tolerated targeted therapy. Beth, maybe you can share some of the learnings from some of that market research. And you may be on mute too.
Apologize. Thank you, David. Yes, we've conducted quite a bit of market research with high volume, high decile practicing oncologists. And what we've learned as it relates to ESR1 is that they are aware of the ESR1 mutation and the fact that it does confer worse prognosis for their patients. And they're very much in need of a drug specifically targeting ESR1. So they currently express the need and the fact that they have a problem, but that they don't have a solution for it. So they would very much welcome a product that is indicated for ESR1 mutated breast cancer, and also a liquid biopsy to easily identify this mutation.
Thanks, Beth. And I do have the questions up now, Glenn. So the next one that we have is, how will lasofoxifene differentiate itself from, as you heard, a fairly crowded field of SERDs, serins, circas, most of those in the degrader camp. I can comment from my experience, having worked on a lot of different SERMs in my clinical research days. And as a practicing OBGYN, I, for one, don't feel that every estrogen receptor is the enemy that needs to be eradicated and degraded. I think that Dr. Salmon's pointed out that these patients who've been on endocrine therapies that have deprived their bodies of estrogen and the tissues that really need to be responsive, such as the brain, the bones, the urogenital tract, all will suffer from complete endocrine blockade. So I think that the idea of leveraging the estrogen receptor, allowing those co-repressors to be recruited, may be why we're seeing the clinical activity that we are with lasofoxifene, as well as not seeing some of the toxicities, both on and off target, that you might get when you indiscriminately, although I think that the SERDs have described themselves as selective, I'm not so sure that they are that tissue specific as the SERM class of drugs are. So again, let me go ahead and first ask Senthil and then Sarah to perhaps add to that.
Yeah, definitely. I think that there's major advantages to using a SERM, and in some sense, the space for the SERMs are crowded, right? This lasofoxifene kind of distinguishes itself being the only SERM that has clinically meaningful activity for ESR1. And as Sarah had mentioned, there are some positive aspects to using SERM, right? So it has positive effects on sexual health in women, positive effects on bone health. And it actually, the lasofoxifene studies have shown decreased cardiovascular risk, decreased risk of stroke, and unlike tamoxifen, it doesn't increase the risk of endometrial cancers. So it's not just the activity of ESR1 itself, but as an agent, it has a very favorable toxicity profile when you compare it to other endocrine agents, including SERDs. So
I would agree, and I would just say that the SERD trials, I think overall in the field, we're all very underwhelmed with how all of the SERDs are performing. I don't think it's all trial design. As you saw, the estrogen population had almost 40% ESR1 mutants, and their PFS in the ESR1 mutant population was only about 3.5 months. So the SERDs are not performing particularly as well as we had hoped. And not only that, the oral SERDs, even though they're well tolerated, a lot of patients wouldn't agree in the clinic. Most of them still have fairly substantial GI toxicity, nausea. Some of them have bradycardia, requiring extra cardiotoxicity monitoring. And those are not going to lend themselves as well to combination strategies that lasofoxifene is. So I think for the reasons mentioned, and those are why I think it's differentiated.
Great. The next question, I think all of us would love to hear some color on, given the excitement at ASCO this year around ADCs in this new population of HER2 low, is the potential impact of ADCs such as Enhertu on the endocrine space. I think, Sarah, you really very nicely pointed out that sequencing is still in those endocrine sensitive patients going to be what you do first. But and I'll let you go ahead and tackle that in maybe more detail. But I also want to point out that while the patients in ELAINE 2 are very different than those that were in Destiny 04, we've had some of our advisors and investigators compare very similar ADC-like activity to the combination that we have, albeit in our small population, demonstrated a 50% ORR in this setting and a PFS of approaching 14 months really hasn't been seen, as Senthil showed in his bar graph, in this targeted endocrine space. So we feel that this combination could confer that kind of efficacy without the toxicity, as well as being oral. I hope I haven't stolen all of Sarah's thunder there, but I'll let her comment further on the ADCs entering this space.
I would just say that it's not really a concern because they're two completely separate categories. So in the clinic, we try to maximize patients' time on endocrine therapy and targeted therapies. And we don't even want to think about ADCs or chemo until we've really exhausted all of our endocrine therapy and targeted therapy options. ADCs are exciting, but they're still given intravenously, and they're still very fancy chemotherapy. They still have much worse toxicity profiles, alopecia, obviously, which is hair loss, neutropenia, TDXD. I'm very excited. I'm on board, but it still has a 10% pneumonitis risk. So that is very much in the IV chemotherapy realm. So we would maximize a strategy like this before we would consider an ADC.
Senthil, anything further to add?
Yeah, I completely agree with Sarah. I think quality of life is really, really important for our patients with metastatic disease. And so there's a lot of value in agents that are oral. As Sarah said, the ADCs still have significant amount of toxicities, including things like alopecia, the risk of pneumonitis. So I think it makes sense to actually look at ADCs almost like chemo-like in patients who have progressed on prior endocrine therapy. And the other thing to point out with Enhertu, as exciting as the data is, I think the testing for 1+, 2+ immunohistochemistry is not very well fleshed out as yet. So there's a lot of variability across labs in terms of testing. So that in itself could pose some problems. Hopefully you would expect that to even out. David had pointed out earlier, in some sense, these are two different populations, right? You're going after ESR1, which would be more endocrine sensitive. And oftentimes when you're thinking about chemotherapy or ADCs, then you're looking at it more in the endocrine resistant population.
Great. Thank you both. So the next question is, and I think that Senthil kind of alluded to this, but maybe we can drill down a little bit more, is that how much do you think that the lasofoxifene contributes to the abemaciclib lasofoxifene combination? The 4 patients that had prior abemaciclib, and maybe we can go ahead and bring that slide up. I don't know if Sarah can go ahead and get that slide. I can actually.
I was able to pull it back. Yeah.
Okay, great. So I think that this slide is illustrative, although again, small numbers. But we had 3 of 4 patients with prior abemaciclib progression who were enrolled. We're grateful that the investigators felt that it was worth looking to see if somehow lasofoxifene could help resensitize patients to that CDK. And our best patient on the swimmer plot at the top of this slide, 40-year-old, very early onset of metastatic breast cancer with high allele fraction of D538G, who had 3 years on her AI CDK, and then a very brief 12 weeks on fulvestrant abema, which is interesting because that's similar to what Kevin Kalinsky's maintain would have indicated that a fulvestrant CDK might have given you in the presence of an ESR1, granted that that was with ribo, and then 7 months on systemic chemotherapy. And now here she is with a longer stable disease at 76 weeks than she achieved with her abema-containing regimen and chemo combined. So I don't know about the rest of the audience, but it seems to me that the only variable we've introduced here is adding lasofoxifene, which I would feel is doing a great deal of the heavy lifting here. In our preclinical data, it did show that lasofoxifene in combination with CDK was the primary driver of tumor reduction, as opposed to with fulvestrant, it's largely the CDK driving that. And then the other two examples of responders, likewise, a very mediocre 16-week response on therapy, the second patient with abemaciclib alone, and then going ahead and enrolling in ELAINE-2 and adding lasofoxifene to the mix gave her a whole different type of response, including a liver lesion that decreased by 71% in a confirmed partial response. So I firmly feel that this, along with the CR that we have confirmed from ELAINE-1 and the activity that we'll present at ESMO, that lasofoxifene has both single agent as well as combination activity and is contributing greatly to this. This is certainly something that we want to confirm in a larger randomized trial. But that is our current thinking that the drug is showing significant contribution to abema in ELAINE-2. So Senthil, please give us your thoughts as an investigator and your experience in the trial.
Yeah, completely agree. I think David summarized it really well. We expect majority of the benefit coming through because of lasofoxifene. So one additional slide I can project is one of the backup slides that we had. And I think that would kind of illustrate the effect of lasofoxifene even better. Let's see. So, it is clear that the effect of continuing CDK4/6 is probably better when you dose them sequentially as opposed to non-sequentially. When you look at the data from the Mass General group, in patients who had abemaciclib non-sequentially, the PFS actually was only about 4 months. But in the patients who had abemaciclib sequentially, then the PFS was about 8 months. So you can see that the effect was largely driven by use of abemaciclib sequentially. We actually have a large preclinical and clinical effort looking at CDK resistance at MDA, and our data so far actually confirms this. So sequential abemaciclib dosing probably is more effective than non-sequential. The reason that is important is because in ELAINE-2, 80% of our patients were non-sequential, right? So as opposed to what we saw before. So the fact that we see such impressive progression-free survival in a non-sequential population with prior abemaciclib would argue that the bulk of clinical response is largely due to lasofoxifene.
Great, thanks for that. The next question is regarding CDX and if there will be a CDX developed for lasofoxifene as a therapeutic, and who are we working with? As was mentioned, we use the targeted panel developed out of Bert Vogelstein's lab at Hopkins, which was commercialized by Sysmex Inostics and now QIAGEN. Which was a very good clinical trial assay. It was fit for purpose for research use. We do know that understanding a broader genomic signature is going to be important, as well as give information about a variety of resistance mechanisms. So we are currently working with Guardant Health. We think that their panel, especially their expertise in liquid biopsies, is optimal. So we are planning on validating and submitting when our therapeutic goes through the NDA process, PMA application for CDX, if there's not one that's already available, FDA approved to identify patients. But as Beth mentioned, I think that the general prescribing community in oncology really like the idea of being able to select patients. And if they're using the Guardant Health panel in their practices, it'll be very seamless for them to see that the patient has an ESR1 driver, and should lasofoxifene be marketed, it would be a very nice way for them to select patients that are likely to have a meaningful response. So we are in that process as we move into our phase 3 studies. Another question is, would lasofoxifene work in ESR1 mutant and wild type? And do we have plans on moving into earlier lines? I know that Sarah showed you data from the Pearl trial, not the Pearl trial that many of the oncology community are familiar with, but the Pearl trial that looked at lasofoxifene, very low doses. In fact, 0.5 milligram was the dose that Pfizer had advanced, but then never launched or commercialized. It's not a drug that's ever seen any kind of commercial availability. And even with that 0.5 milligram of lasofoxifene, you saw a very impressive reduction in de novo first cancers, which we know by definition will be wild type. That these are patients that have osteoporosis. They haven't seen any endocrine therapy. So the assumption would be that all the breast cancers that were prevented are essentially subclinical cancers that were essentially treated by low doses of lasofoxifene, preventing them from becoming clinically relevant. So that and our preclinical data, as well as some early preclinical data, certainly show that, like a good SERM, and we think we have the best SERM, that it has activity not only in ESR1 mutations, but also wild type. I don't know, Sarah, if you want, or Senthil, want to comment further, at least on this issue about wild type and ESR1. Paul Pleward, who's also on the call, always reminds us that none of these tumors are heterogeneous. So we know that the agent has to act across the board on both the ESR1 mutant and wild type cells. So maybe Sarah, then Senthil, can talk a little bit about SERM, SERDs, and wild type and ESR1 activity. Any additional thoughts, Sarah, and then Senthil?
I think we already talked a little bit about why I think, SERMs and ESR1 mutants are exciting. Just commenting a little bit on whether or not I see this coming in the earlier stage setting. I think this drug positions itself well to be tried at some point in the adjuvant setting, and I would actually see it placed in an aromatase inhibitor sort of intolerant population. So the standard of care therapy for women with hormone receptor positive early stage breast cancer who are postmenopausal is an aromatase inhibitor. But nearly 40 to 50% of those women do not tolerate aromatase inhibitors due to hot flashes, cognitive difficulties, issues with sleeping, bone loss. So that is actually an extremely high need area to see a compound like this that improves bone health, that has activity in breast cancer prevention, that apparently potentially improves cardiovascular health. I mean, that's another huge concern in the adjuvant setting. So I actually see this drug positioning itself well eventually in an adjuvant AI intolerant setting.
Yeah, I completely agree with Sarah. I think this drug has a lot of value in the adjuvant setting, considering its favorable risk and benefit profile in terms of women's health. So I think there'll be a lot of interest in future in terms of applying this medication in the adjuvant setting. And one thing I would add is, with abemaciclib approved for high-risk early-stage breast cancers, if there is actually synergistic activity for laser with abemaciclib in future, you could actually see a role for laser with abema in high-risk breast cancer patients early on.
I totally agree. Yeah, I agree there.
Yeah, no, I think that those are great points. That the persistence on AIs is unfortunately very low. We know how well they work when patients stay on them for 5 to 10 years, but that's an awfully high bar to ask anyone to have that kind of estrogen deprivation, particularly in these young patients, for with a decade or more of depriving them of estrogen. I know, Beth, that we talked a little bit just about how ESR1 targeted therapies resonated with the high prescribing physicians that we've done some market research on. There's a question here about the size of the market share. We're a small earlier mid-stage biotech, but we're very proud of the kind of market research that we've done that's congruent with our clinical strategy. Beth, maybe you can comment, at least on a high level. I know we've got a huge amount of information about how our product profile resonates with prescribers. Maybe you can describe a little bit of what we've learned in that regard.
Thanks. Yes. As David mentioned, we have continued to pressure test this market with very robust and granular market research to see how the market is evolving. And what we have learned is that the high prescribing oncologist is very comfortable. They're very comfortable with SERMs. It's a class that they know and that they trust. And what they would like is a more effective SERM. With respect to product attributes that are important to these physicians, they are looking for PFS first and foremost, and then quality of life has moved into number 2 attribute for physicians. So we've seen that over the past 3 years really move up in terms of importance. We also know that the ESR1 patient population is a very easily identified and targeted addressable patient population, with a liquid biopsy test. And so this brings reassurance not only to physicians about who to treat with this targeted precision medicine therapy, but also on the access side with payers, that they know that that a drug that is prescribed will work in that particular patient population. So some of those access and payer hurdles have been diminished. So with respect to the market, again, an easily identified population, a drug that we believe is best in class as an endocrine therapy, at least shaping up to be so on the PFS side, certainly on the quality of life side. And when we take a look at that addressable patient population and peak market share of about, peak is up to almost 50 to 70% within that ESR1 population, our forecasts are looking like they're going to head into $1 billion. For peak share, which you've probably heard for some of the other endocrine agents as well. I would like to also add on the market research side that patient and the patient voice is becoming more and more important, I think, as Sarah and Sentil have also alluded to. And we are conducting a patient survey called Equals. It's the Elaine and ESR1 Quality of Life Survey with an expert group of very esteemed oncologists as the steering committee. Dr. Simons is one of those, and we are planning on submitting that survey into San Antonio. So hopefully we'll be hearing more in the coming months, as it relates to the patient's impact and quality of life concerns, as well as knowledge about ESR1 mutations.
Thanks, Beth. I'd like to bring Dr. Paul Plourde into the conversation, given his extensive experience in working in this endocrine space, right? As a key leader in the fulvestrant, anastrozole, and tamoxifen programs. Paul, maybe you can just share. We've been working together for quite some time. Maybe you can just share some of your experience and your thoughts about working on the lasofoxifene program and how it looks vis-a-vis what was done in previous research at your time at AstraZeneca.
Yeah, thanks, David. Maybe we've been together too long. We've both gotten grayer, but the experience with lasofoxifene has been outstanding. As David mentioned, I worked in the hormonal field for quite a few years at AstraZeneca, and in particular the SERM tamoxifen and subsequently fulvestrant. And the results that we're seeing this early, and so specifically on a population that has such a strong medical need, is just uncomparable to the experience I had with those other compounds. To see a complete response with this particular agent, and then to see the PFS in Elaine 2 be 13 months with 50% objective response rate, almost unheard of. Certainly, we did not see that with tamoxifen and with fulvestrant. So this compound, as we develop it further during our registration period, really is a cause for a lot of optimism for patients that they would get a treatment that would be so beneficial and so well tolerated, that I think for physicians, once we get this on the market, will be excited to have it.
Great, thanks, Paul. And you're right. We've added a few gray hairs these last few years. Another question is that and you kind of point out to these robust results of lasofoxifene and abemaciclib in Elaine 2. But I think that Senthil really pointed out, and we kind of addressed, the potential contribution of laso to that. Question is that, would you anticipate that lasofoxifene with palbociclib or ribociclib would work as well as a combination with abema? And then why did you study it with abema as the CDK? I'll take that second part, and I'll maybe let Senthil add some. I think he already kind of described the differentiating characteristics of abemaciclib. But it made a lot of sense given, and as you saw, the distribution of prior CDK. I think this may change, particularly after the OS data that was revealed at ASCO with palbociclib. But given that most patients had seen either a palbo, and we thought maybe a little bit higher share for ribo, we didn't see that it made sense that clinicians would be more comfortable sequencing with a different CDK. And given that abema had a monotherapy indication in later lines, we thought that perhaps it was the more potent of the CDKs. I think there's still a lot of work that needs to be done about how these differentiate. We're certainly glad that Lilly was amenable to a collaboration in supplying abemaciclib to this very positive and reassuring result with a combination. And my guess is that based on our preclinical data with palbo, I think laso and palbo or laso and ribo would work well. We have the data we have, and we think that this combination may be doing something unique. We'd love to explore it preclinically and see if there's other signaling that's happening between these two combinations and some potential crosstalk that might be unique. But let me ask maybe Senthil, since he gave you a little bit of information on that, and then Sarah to add further to whether or not we've stumbled across a very unique combination, or it could be CDK agnostic.
Yeah, I think as David summarized, I mean, it's largely dependent on the question that we were asking and the population we're looking at, right? So we were looking at clinical activity for patients who had progressed on post-CDK4/6 base, and we were looking at partners that included PI3 kinase inhibitors versus drugs like abemaciclib. And preclinical studies have shown that abema still has activity when patients have progressed on palbo and ribo. And in fact, we are actually planning to complete a paper at MDA where we looked at preclinically giving palbo for patients who had progressed on abemaciclib before. They do not respond, but the reverse is true. So there's, as one of the reasons to pick up abemaciclib in post-palbo ribo progressed population. But having said, I don't see any reason why laser cannot work well with palbociclib or ribociclib. But again, it depends on what line of setting you're actually looking at, right? So if you're looking at ribociclib or palbociclib, then in a sense, lasofoxifene becomes the first line agent almost, because you're looking at it in the first line metastatic setting. So one thing to be cognizant of, like, because we're also enriching patients with ESR1 mutations. So these mutations evolve, more you actually get endocrine therapy. So if these patients are being studied in de novo or first line setting, the enrichment of ESR1 mutation is not going to be that much compared to patients who had progressed on prior therapies.
Yeah, great point. Sarah, anything further to add on selecting a CDK? I know it's a area of debate.
No, I would agree. I mean, the only other thing that I'll add is there's a little bit less enthusiasm, I think, overall for palbociclib given the Paloma-2 data. So I think ribociclib has positioned itself quite well moving forward as the preferred first line CDK4/6 inhibitor. That's certainly not going to be true for everyone. Not everyone feels that way, but I think a lot of us feel that way moving forward. And abemaciclib, we haven't seen their overall survival data yet in the first line setting, but abemaciclib is the only approved adjuvant therapy. And so we're using that in the high risk adjuvant setting, but abemaciclib is probably going to be the most successful CDK4/6 inhibitor for use after progression on a CDK4/6 inhibitor. Like Senthil said, palbo is not going to work after CDK. Ribo, the maintained data was not super impressive, but because abemaciclib has that CDK2, CDK9, broader depth of inhibition, it's positioned itself very well to be the best second post-CDK.
Great, thank you. And then the last question I have here, and if there are others in the audience who haven't entered any questions into the the chat function, please do so. We've got about 9 more minutes. Is what other combinations might prove beneficial in this setting? We feel that given lasofoxifene, and we have 27 phase 1 studies, courtesy of the prior development program, is that there's little to no DDIs that have been identified with lasofoxifene. So we feel very comfortable that it could be an endocrine backbone of choice with any variety of targeted therapies. Our focus is clearly on the very strong signal that we see here in the post-CDK setting with the ESR1 mutant-driven cancers, but there's certainly the potential to pair this with an mTOR inhibitor, such as everolimus. It could be looked at in a coexistent population, both with everolimus that have a ESR1 and a PIK3CA mutation, as well as possibly alpelisib with a coexisting PIK3CA mutation, which overlap with ESR1 mutants, by the way, about 30% of the time. And we've seen that in our trials as well. And we did see some nice responses in 2 out of 3 of patients in ELAINE-2 who had progressed after prior PIK-RAY. So we know that it does have, even without an mTOR path inhibitor targeting specifically the PIK3CA mutations, that laso-abema had activity. But it would be very interesting to look, I think, at laso with those drugs targeting that pathway. I think there's some excitement around AKT inhibitors. We'll see if that's a potential partner to pair with lasofoxifene, as well as potentially dual endocrine targeting therapy with something that might hit the androgen receptor as well. So those are all things that we're actively exploring. I think that our advantage is compared to some of the oral SERDs, which we've seen can have some significant DDIs. We know that the Zentalis program in combination with CDKs was terminated because of DDIs, and we know that amcinestrant in higher doses reduced exposure to palbociclib significantly, and even at the selected phase 3 first line study with palbo, they're still going to see a 20 to 30% reduction in exposure. So we feel we're in a very good position to have lasofoxifene emerge as the endocrine backbone of choice with a variety of therapies and a variety of lines of therapy. So we're very excited to move the program forward and look forward to this year as we gain further clarity on our first path towards registration. But certainly, given the right partners, the right capital, we think that the sky's the limit for this very versatile small molecule that's so well characterized. That was the last question we have. Maybe just let me, in the last 5 minutes, unless there's a late breaking question here, maybe ask Sarah and then Senthil just for some summary thoughts about their experiences. I know they gave us eloquent presentations, and we really do appreciate their partnership and their work on their respective trials, the ELAINE 1 and ELAINE 2. We've had some terrific leadership from them, as well as our other investigators, which include folks at the Cleveland Clinic and Mayo Clinic, as well as many others. So maybe just to wrap things up, Sarah, maybe you can comment on your feelings around ELAINE 1, going to report out here very shortly, and then let Senthil wrap it up with some concluding remarks.
Yeah, it was a joy being a investigator on ELAINE 1. I think offering an endocrine monotherapy in a very select population was just fun to accrue to. In my patients that received the drug, they had the absolute best quality of life. And we really can't minimize what that means. These women on average live for more than 5 years. So we really are thinking a lot about their quality of life, and patient-reported outcomes in registrational trials are now standard, because it's extremely important to not only look at toxicity, but how a patient feels about that toxicity. And I don't know of another drug that actually makes a patient feel better from a vaginal and sexual health standpoint, which is a huge problem with endocrine therapies. So I love that part of developing this drug. It's tolerated very well. They've been successful to date, I think, because they're focusing on an ESR1 mutant population. And then we'll just have to wait until you see the ESMO results.
And Senthil.
Yeah, so if I can add, so we had met with David, I think Paul and Miriam in San Antonio 2019. We were looking at partners or like agents for ESR1, and COVID-19 hit, but they were kind of focused, relentless, and moved forward with ELAINE 2. And they're an absolute joy to work with. They're very thoughtful. They think about patients. So it is a great, collaboration that we have had. And in terms of the activity of the agent itself, we've not seen this kind of response rate or PFS with any endocrine therapy combinations that I know of. And as David had alluded, this is kind of the response that we see more in the ADC space, or like, so this is really, really encouraging. So we look forward to next steps in terms of how to move this forward. And finally, from a physician's perspective, in terms of enrolling for patients with ESR1, at MD Anderson, we have multiple biomarker or genomic trials at MDA. I think we probably have more biomarker or genomic trials at MDA than anywhere else in the world. But I can tell you, like, out of all the biomarker, targets or enrichment, this study had the most interest. So I think everybody wanted to look or wanted to put patients on ESR1 targeting agents. So I think that kind of speaks for the strength for ESR1 as an unmet need. And also with the recognition that we've seen at ASCO and from patient care advocates from GRASP Foundation and others, that again speaks for how ESR1 is an important target.
Great. I want to thank you both for your leadership, your partnership, helping us accrue these two very important trials, which we feel are only the beginning for lasofoxifene. That kind of takes us to the bottom of the hour. So we want to thank everybody who called in, asked questions, and if you need any follow-up, Glenn and Sarah are certainly available to receive email if you need to be put in touch with either of our speakers or anybody from our team. We look forward to continuing to inform you about our program's progress and hopefully continue to get this information out into the public and in the literature. So with that, let me again thank everybody, thank LifeSci for organizing the call, and wish you a great rest of your day.